And so our life is not predetermined by the genes that we inherit from our parents. They are important, and as the father of identical twin daughters, anyone knows that identical twins are remarkably physically similar, though I can tell you their personalities are quite different. And of course our environment has an enormous effect on things like cognition. So this is a crude depiction of the sort of gradient between environment and genes in the causation of disease. So at the bottom, we've got a disease which was really the first inborn error of metabolism to be characterized, described by Garrett in his 1908 Croonian lecture to the Royal College of Physicians. And this is associated with urine that turns black on oxidation. And it's also known as ochronosis because it has a yellow and ochre-coloured pigmentation to the tissues. It's a relatively benign condition, though it does cause severe arthritis with aging. And that's pretty much 100% genetic. If you inherit both those alleles from your parent, then you will get this disease. At the other end, there's infectious disease. You quite clearly won't get flu unless you're exposed to flu. You won't get tuberculosis unless you're exposed to it. But actually in the 1940s, it was recognized that there's a significant genetic component to tuberculosis. So if you look at the frequency of tuberculosis in a pair of identical twins, it's much higher than the coexistence of the disease in non-identical or fraternal twins. And then in the middle, we've got diseases like type 2 diabetes, where clearly what you eat, becoming obese, is a very important component of diabetes, but also there are important familial factors as well. And so there's a range of influences varying from mainly genetic to mainly environmental, but at either end, both factors are important, and it's important to recognize that. And one shouldn't really get into a fight about nature or nurture. It's not about fighting, it's actually about discovering the truth. And so here's the example of obesity. And obesity is something one thinks of as being incredibly environmental. But this is a study from 1976. And this shows the power of twins separated at birth. And indeed, my PhD mentor very kindly suggested that we ought to separate our identical twins at birth because it would be experimentally more interesting. We declined to do that. But at the bottom, you see monozygotic twins, so identical twins, who were separated at birth and photographed many years later. And you can see how extraordinarily similar they are, though they've been brought up in different environments. At the top, you see dizygotic, so fraternal twins separated at birth. And like any other pair of brothers or sisters, you can see that they are extraordinarily diverse in their appearance. So actually, obesity has quite powerful genetic components. And indeed, this is an example of an extreme, very rare form of monogenic obesity. This is caused by a mutation in a single gene called leptin. And this is a good news story because the leptin has been cloned, and the image you see on the right is of the same child who's been treated with injections of leptin, which has suppressed the child's appetite, and you can see that he's now essentially normal. Now, of course, it's one thing to discover the gene, it's another thing to work out what it does. And so we really are, with genetics, at the start of an enormous medical adventure. Because actually, for every gene that's discovered, one's got to work out how it causes the disease, and then having worked out how it causes the disease, whether there's anything one can do about it. And this is the sort of study that you can do if you're interested in, for example, genes and appetite. And so Sadaf Faruqi and Paul Fletcher, who have been two Wellcome Trust Senior Fellows, have done functional brain imaging, and that's the sort that Dermot Kelleher showed you just before. And you can see on the bottom 3 MRI images of the brain, and you can see little coloured spots, one of them circled in red. And those are areas of the brain that are activated when someone in a scanner is shown a comparison between, on the top, for example, food, and on the other side, something which I think most of us agree was not food. If you were a car enthusiast, your brain would react in a different way. And on the bottom, you can see what, well, some might consider on the left to be appetizing food, and many would consider bland food, the broccoli on the right. Now, if you're a patient that has leptin deficiency, then your brain is activated to both the hamburger and the broccoli equally. But after injection of leptin, then they no longer get brain activation to broccoli. They behave like most people in this room would, which is activated to hamburgers and not broccoli. And you can start seeing on the bottom the examples of the changes in brain activation and blood flow pre and after treatment with leptin. So what these studies show is that the genetic influence that controls the susceptibility to obesity is actually in the appetite control part of the brain. And that's turned out to be incredibly important. So most of the genetic variants associated with difference in body mass index with obesity are genes that control our appetite. And indeed, if we took everyone in this room and afterwards, with the banquet that we're all going to be offered, you compared how much people ate, the people with greater susceptibility to obesity genetically will eat more on average than those without, although none of us actually know which genes we have. Now this is important for other reasons as well. So this is another monogenic, so single gene, cause of obesity. It's quite difficult to see this, but there are two grey columns you can see. And on the grey column on the left is a genetic area where there's a gene that controls appetite. And on the right, there's a gene associated with autism. And so if you're an individual who's unlucky enough to have a DNA deletion, a bit of DNA removed, that affects both those regions, then you'll have both obesity and autism. And if you have a mutation which is associated with the left one, then you'll have obesity, the right one alone, you'll have autism alone. Now, this actually turned out to be very important, because one of the families of children here was put on an at-risk register by social services because it was thought that their parents were abusive. And actually there are several examples when this has happened. There was a terrible case when a very obese young child was plastered all over the front page of one of the tabloid newspapers. And it turned out that child had a mutation, I think it was in leptin or a related gene. And so it's very important to make these diagnoses, because even if it's necessarily a treatment, it stops society doing awful and wicked things to the parents inappropriately. Because children who have leptin deficiency, you basically have to put locks on the fridges, you have to put locks on everything. They will eat and eat and eat because the drive is so great. Those were some monogenic disorders. This is an example of one of those studies I told you about. They're known inelegantly as GWAS studies, which stands for Genome-Wide Association Studies. And what you see here, colour-coded, are the different chromosomes from 1 through 22. And each of those columns is in fact many, many different genetic variants. And of course they just fuse into one at the bottom, there are so many. But what you can see are two peaks. One in the middle called TCF 7L2, and one on the right which is called FTO. And it turned out that the FTO variant, genetic variant, is associated with a tendency to obesity, and through that is associated with an increased risk of type 2 diabetes. And when this was discovered, it got the most extraordinary publicity. For example, scientists find the gene that makes you fat, and I have to say it isn't the gene that makes you fat, it's the eating that makes you fat. But these genes, which are the variants common in this population, they do have a significant influence on body weight. And so here are two on the right. FTO I've talked about, and the other one is called melanocortin 4 receptor, MC4R. And if you have the variants associated with increased weight in both MC4R and FTO, then on average you will be about 3.5 kilograms heavier than people that don't have those. And 3.5 kilograms is a reasonable amount of weight. Now, again, the challenge is to understand the physiology. And this is where genetics comes together with cohort studies. And the challenge and the opportunity for the future is to marry what I would call phenotype with genotype. So phenotype is our appearances, every aspect of our appearances. It's about studying diseases in the great detail so that you can understand all aspects of what makes them different. Obesity, body mass index, is a phenotype. So the question with this was, well, when did it start having an influence? Was it an influence on fat mass or on lean mass? And the Wellcome Trust and the Medical Research Council have collaborated for many years to fund a longitudinal birth cohort of children who were born in the Bristol area in 1992. It's called ALSPAC, the Avon Longitudinal Study of Parents and Children. And it was possible in a very short space of time to go from discovering that FTO variants were associated with obesity to look in the ALSPAC population where all the samples were stored, all the consents were given. And you can immediately see that on the top of this graph, the children are 7, 9, and 11 years of age. And you can see the red bars, and this is body mass index, are associated with a higher body mass index than people that have a mixture of the variants, an A and a T, and people in blue, who are the individuals who have the variant of FTO, which is associated with lower body mass index. So you can see there's already an influence at 7. In fact, there was no influence on birth weight at all. You couldn't distinguish these babies on their birth weight. And at the bottom, it was already known whether their fat mass versus their lean mass. And you could see immediately that the FTO effect is on fat mass and not on lean mass. So by bringing together genetic variation with phenotypic information, you can discover things extraordinarily quickly. And actually, I have to say, and we're a research funder, paying for this study from scratch would have cost a fortune. But actually, it cost virtually nothing, this particular add-on, because it was a marginal cost on a study that we were already funding. Now, I'm not going to take you through this slide in detail, but the point is, this is now looking at the genetics of type 2 diabetes. And what you suddenly find is that the genes that are associated with the increased risk of developing type 2 diabetes cluster into physiological pathways. So I've already talked about obesity, which is a risk factor. PPAR gamma is associated with insulin resistance. But on the right, you can see beta cell dysfunction, so the beta cells are the cells in the pancreas that produce insulin. You can see reduced insulin secretion, you can see reduced beta cell mass. So a lot of the genes are in physiological pathways that affect the activity of the pancreas. And indeed, for many years, there'd been a bit of an argument about whether it was a problem in the pancreas or it was all peripheral insulin resistance. So resistance to the hormone which regulates glucose metabolism. And it turns out that actually it's mainly dysfunction of the cells that manufacture insulin, all discovered by this form of genetic analysis. Now, another thing that's talked about a great deal is pharmacogenetics. That's using genetics to influence prescription to work out which drug works for which patient. Now, we're a long way from being able to do this at scale, but there are already some quite interesting examples. This is a rare example, but an important one. So this is a group of children born with a severe hereditary form of diabetes. And these children were treated with very large doses of insulin, and it didn't work very well. Now, a bit of genetic analysis showed that the mutation in this disease was in a potassium channel called inelegantly KIR 6.2. And when that was discovered, it was realized that this channel is a target for drugs that are used in diabetes called sulfonylureas. And so Andrew Hattersley and Fran Ashcroft, working in Plymouth and Oxford, basically, it was Andrew Hattersley, who was a clinician, Fran Ashcroft, who was the cell biologist, switched these children from insulin to sulfonylureas with the most dramatic effect. And these children now have their diabetes well controlled on sulfonylureas. So this is an example of using genetics to influence prescription.